Hello, everyone. My name is Paige Chesshire, and today I'm talking about the scientific name cleaning process that we used for our USB data completeness project. We all know that occurrence data are critical for conservation and research purposes. This includes specimen and observational records. Online databases like GBIF and SCAN are invaluable, and other platforms like iNaturalist and Bug Guide help mobilize community science data. These occurrence records can be used for spatiotemporal analyses on various scales, but the data does need to be standardized, especially the scientific name columns. And so there's often extensive cleaning that does need to occur before analyses. Our USB completeness project was published in Ecography earlier this year. The goals of this project were to conduct data completeness analyses of bee assemblages at 4 spatial scales. So here on these maps, the top map is bee data completeness for all 6 families in the US at a 30 kilometer by 30 kilometer resolution. And then the lower map is completeness at a 220 by 220 kilometer resolution. And the point is these bee data across the contiguous United States is still very incomplete. We also had an additional element where we knew from a survey done by iDigBees across many US institutions that there are at least 4.7 million additional US specimens in US collections that could be unique and digitized and added to our data set. So we projected 4.7 hypothetical data points, added those to our data set, and we found that even when we projected data completeness with an additional roughly 5 million records, the data biases that we see now will still persist. So we do need to initiate new strategic surveys, and we do need to target undersampled taxa and locations. Our methods for this involved creating range polygons for 3,158 USB species. But before we could create these polygons, we did need to clean the downloaded data. We couldn't just use the scientific names as is. If we would have tried to run species ranges by grouping scientific name, it wouldn't have worked because the scientific name column looks like this upon initial download, with a lot of extra characters and dates and author names. So for our project, we started with 2.962 million North American occurrence records that came from GBIF and SCAN, as well as a few additional CSVs of bee data. Steps 1 and 2, we just removed duplicate records across data portals using duplicate combos of catalog number and institution code. And then also we did remove a few records that were completely lacking scientific name. But that was really only 3 records because most records had something in the scientific name field. Step 3 was just a very initial name cleaning where we kind of eliminated parentheses, author, year, extra characters to try and get a more concise list. And then step 4, we identified records that had imprecise latitude and longitude, which was about 478,000 records. 296,000 of those were considered imprecise lat longs, where the coordinates maybe only went to 1 or 2 decimal places. But then there are 182,000 that were completely lacking coordinates. So we recruited Nelson Rios to use his geolocate tool to get better coordinates for some records. Next, there were issues with the date field. So I sent the data to a co-author, Erica Fisher. She made the event date column uniform and filled in the year or day if they were missing in those columns, but that info was available elsewhere, maybe in the verbatim date column. And then we ran the list of scientific names that we had kind of gathered up until that point against a SCAN taxon table to collapse subspecies and drop extra words. And this same method removed any additional dates or numbers or characters that were still persisting in that scientific name column. Finally, step 7, we actually start removing big chunks of records. So we removed 2,900 records that were still missing coordinates, even after Nelson Rios tried to find some. We removed 166 records from families that aren't even bees. And we removed 372,000 records that were single name scientific name, meaning they weren't ID'd to species, and similarly, we removed morphospecies. Step 8, this is where things got a little more complex because it was a mix of R scripts and manual name changing in Excel and reading spreadsheets back in and using a lot of outside perspective. So for part 1, we ran the clean taxa list that we had, gathered up until that point against the Discover Life synonym list. So the Discover Life Global Bee Taxonomy list is created and run by John Asher and John Pickering. They are the leading source of global bee taxonomy, and this list is peer reviewed by global experts on an ongoing basis. So using this Discovered Life taxa list, we determined which scientific names in our list of names were accepted already, which ones were considered canonical and needed to be changed, which ones were not found in either the accepted or canonical columns. And then we had to kind of output those names and make manual decisions on what to do with each one. We then also compared our list of names that we'd gathered up until that point against a accepted list of North American names that GBIF has. And we still identified 372 names that don't match the taxa in the GBIF list. So often they were male or female discrepancies. Sometimes the species we had were not known to inhabit North America, but the point is that we had to check each of these names manually and make a decision. Do we keep this record or remove this record? I then compiled the results for all those previous spreadsheets to create an absolute final species list. And then I read in that species list to R. And if it was applicable, I told the new name to replace the original scientific name. Ultimately, I produced a clean data set of 1.923 million contiguous USB records for 3,158 bee species. And this data and code are provided in our Figshare and GitHub repositories, both of which are linked to the ecography manuscript. But as you just saw, some chunks of the cleaning are still not 100% reproducible and require individual eyes and opinions and manual changes in Excel spreadsheets. So while this was a giant undertaking, there are still some things that I would like to be more reproducible. And so ideally, all of this can happen using R scripts alone. Data cleaning R packages are ideal. And a thorough biodiversity data cleaning R package does exist. It's known as BDC, created by Ribeiro et al. Last year. But the next thing I want to talk about for the last couple minutes here is a collaborator, Dr. James Dory, has built upon BDC to create BDC, which is a package specific to bees. And this project incorporates the same type of cleaning steps that we and others do for these types of papers, like identifying spatial, date, and taxonomic issues. And the functions that he wrote are a combination of his own work and BDC functions. But what's really unique about this package is more aspects of flagging and cleaning the data are reproducible with R, and those problematic records are flagged, but they are not discarded unless you make the choice as a user. So step 0 through 2 are script and data preparation. Data was sourced from GBIF, SCAN, iDigBio, USGS, as well as some other smaller public or private data sets. Step 3 is initial flags, where records are flagged with initial obvious issues, like scientific name flags, or records that are missing coordinates, or coordinates and country don't line up. Step 4 is the taxonomic cleaning part. So again, the Discover Life list by Asher and Pickering is used. And in this step, names are clean, extra characters and numbers are removed from that column. But then also this really cool custom function known as Harmonize R matches Discover Life taxonomy with the list of names in the data you are using. And so this is similar to the manual name cleaning that was done in the USB paper that I led, but it allows for more reproducibility. Step 5 is space flagging, where problems with coordinates are identified, like coordinate uncertainty or coordinate precision issues. Step 6 is time flagging, where either records are either recovered or filtered out if there are date-related issues, like missing event date or extracting the year from the event date column. And so, again, this is similar to the year cleaning step done by my collaborator or co-author, Erica Fisher, but it is all done in R in one package. So another really cool thing about this is for steps 3, 4, 5, and 6, all of those records that were flagged are saved as separate data sets for evaluation by the user. So the user can go through and say, all right, these were flagged, but I don't necessarily want to discard them. I want to look at them further. And then step 7 is just very quickly removing duplicate records, which needs to be done for all of these types of data analyses papers. And then step 8 is a final filter of taxonomic cleaning, and it provides code for users to produce a clean data set to their own specifications. So you can download each of the flagged data sets. You can rerun specific functions on certain subsets of the flagged data sets. This package is also set up to run very specific queries on the full data set or flagged data sets. And ultimately, the full script will produce a fully clean data set and an uncleaned but flagged data set. So this package is going to be really, really great. And we are so grateful to James Dory for leading it. He's really been working so hard on all of the functions. The code is currently being test run by numerous collaborators. And the R package and the associated data paper is under review at Scientific Data. And this package really will be huge for cleaning bee data across regions. So we're excited for it to be available, hopefully in the next few months or this year. So thank you very much.